cardiovascular diseases remain as the leading cause of death worldwide, claiming approximately 18 million lives annually. This made me wonder, what if the key of preventing it lies right in our genes? In this video, I'll be using AlphaFold to depict how the ACE enzyme looks, which is a component of regulating blood pressure, and how a genetic variant called the ACE insertion deletion polymorphism interacts with it, leading to hypertension. First of all, what is hypertension? Hypertension is a condition where your blood pressure reaches to unhealthy levels for a prolonged period, and this ultimately leads to the development of heart diseases and life-changing incidences like a heart attack. Hypertension is a common disorder affecting almost 1 billion people worldwide. To put that into perspective, almost 1 in 4 people are affected by it. At the very center of this issue lies a critical enzyme known as the angiotensin converting enzyme, also known as ACE. And it is a key component of the renin-angiotensin-aldosterone system. I know it's very long, but you can call it RAS, which regulates your blood pressure. But before we really get into the science behind this, let's start off by depicting ACE on AlphaFold. We'll start off by using a resource called UniProt, which is used to access the protein sequence through their database. We'll begin by typing in the name of our protein, which is called ACE. Okay, the first entry is the one that we'll be using, as the sequence is based off of Homo sapiens, us humans. So right here. We can also see that this protein comes from the ACE gene, and in terms of function, it plays a major role in the angiotensin-renin system that regulates blood pressure and sodium retention. To access the amino acid sequence, we could just click right here. And so the amino acid sequence of this protein is quite lengthy, as it is composed of 1,306 amino acids. The length of each protein varies as amino acids determine the complexity of it through the functions these proteins are able to perform. So here we will just copy the sequence, and from this step forward, we will be transitioning on to the AlphaFold server. Since I already had the protein pasted, I'll just click Continue and Preview Job, Confirm and Submit Job. And so this process only takes about 5 to 10 minutes to model the protein, so just hang in there. Since I already had the protein modeled from beforehand, this is what we'll be referring to. And so to start off, the predicted template modeling PTM score shown at the top is 0.75. This shows that it was a fairly decent prediction. A PTM score above 0.5 means that the overall predicted fold accurately resembles the true protein structure. We can also see that 4 different colors are used to assess the accuracy of our complex. Dark blue, which has a confidence score of greater than 90%, surrounds pretty much most of our protein. Cyan, which has a confidence level between 70 to 90%, takes over just a small portion. And yellow, with the confidence level of 50 to 70%, is just a sliver of our protein, so right around here and here. Lastly, orange, which scores a confidence level of less than 50%, can mainly be seen at the 2 ends of the ACE protein. And at the right, there's a graph showing the scored residue compared to the aligned residue. This represents the predicted aligned error, which is a measure of how confident AlphaFold is in the relative position of the residues shown in the predicted protein. Residues are when 2 amino acids bond with each other. So in the plot, the scored residue refers to the residue being evaluated for positional accuracy and seeing if they're binded correctly. On the other hand, aligned residues is the one being compared to the chosen residue. So for instance, if we take the scored residue of 1044 and compare with the aligned residue of 783, the coordinates land in the dark green square. This tells us that the positioning of the residue is highly accurate. Lastly, at the bottom of the plot is the expected position error, measured in angstroms. This represents the model's predicted uncertainty about the relative position of the residues in the protein structure. Angstroms is a unit of measurement used to determine and measure nanoscopic distances like the ones in proteins. So these are pretty much the steps of modeling the ACE enzyme on AlphaFold. Now that we've modeled our protein, we can use our learnings to understand what the RAAS system does and how it connects to the ACE enzyme. RAS is a complex hormonal pathway essential for regulating blood pressure, as I mentioned before. So this system kicks off with the kidney. So when your body notices that you have low blood pressure, the kidney releases renin, a type of enzyme. And so the renin acts on the protein angiotensinogen, and it creates angiotensin 1. Angiotensin 1 is then transformed into angiotensin 2 when it interacts with ACE, the ACE enzyme which we just modeled on AlphaFold. So angiotensin 2 is the primary hormone in this process as it raises blood pressure levels by causing blood vessels to contract, or in other words, get smaller. And in turn of this, aldosterone, another type of hormone, is released from the adrenal glands, which are two glands sitting on top of your kidneys. And aldosterone then prompts the kidneys to retain more sodium and water, elevating blood volume and pressure. So, in a healthy RAS system, this mechanism adjusts itself to maintain normal blood pressure. However, with those who have hypertension, their system becomes more overactive. And this overactivity is caused by chronic stress, high salt intake, or even genetic variants like the ACE insertion deletion polymorphism, which is what we're focusing on in this topic. The ACE insertion deletion polymorphism is a variant within the ACE gene. It is characterized by the presence, represented by the letter I, and the absence, by the letter D, of a 287BP allorepeat sequence. Now, what is an allorepeat sequence? In simple terms, it is a highly repetitive DNA. What is extremely fascinating about this polymorphism is that it results in 3 different types of genotypes, or genetic makeups, that being DD, ID, and lastly, II. These differences can influence the expression of the ACE gene. After reading through several research papers, I came to the conclusion that the DD genotype affects the ACE enzyme levels the most. So this means that there will be a greater conversion of angiotensin 2 hormones, which then lead to increased blood pressure. The ACE-ID polymorphism could help with future research in countless ways. Firstly, it could guide more targeted therapies, increasing the efficacy of hypertension treatment. It could also serve as a genetic biomarker for identifying those with a greater risk of having hypertension, basically those with the DD genotype. Recently, I had the incredible opportunity to meet with Dr. Perret at a McMaster University research lab. In regards to cardiovascular genetics, he proposed that the risk of heart disease is not just because of one single genetic mutation, it's of hundreds, in fact thousands of weaker variants that are causing it. So with that said, the ACE-ID polymorphism might be just one variant that results in cardiovascular diseases. Although I wasn't able to show the direct impact that the ACE-ID polymorphism had on the ACE enzyme, I was able to meet with Dr. Perret at McMaster University and simulate how the ACE enzyme looked on AlphaFold. These learnings have expanded my knowledge behind the science of cardiovascular genetics. I hope you learned something new, and thank you for watching. Bye!